Imagine for a moment this hidden, ancient system deep inside your body. It's like a silent army, really, over 50 proteins, always on patrol.
Yeah, constantly working.
And it's doing way more than just fighting infections, which is, incredible on its own. It's also cleaning up cellular junk. It's actually shaping your brain. And get this, it's involved in diseases you might never connect to immunity.
It really is a remarkable network. And, what started over a century back is just this simple observation, something that seemed to, well, complete what antibodies were doing.
Right.
That idea has just blossomed. Now we see it as this incredibly complex, multifaceted system. It operates right at that interface, between our immediate innate defenses and the more tailored adaptive immune response.
Exactly. And that's what we're diving deep into today, the complement system. You've shared some really rich, detailed sources on this cornerstone of our immunity. And our mission really is to unpack every key concept, every surprising detail, so you listening can get a truly deep understanding. Like the top experts in the field grasp it. We'll dig into its history, those intricate pathways, its many, many functions, how our bodies control this powerful thing, how microbes try to fight back.
Evasion strategies, yeah.
Its deep evolutionary roots, and even how we're using this knowledge now in medicine. We really want to make its complexity clear, compelling, help you master this.
Sounds great.
So let's lay the foundation.
Right.
When we say complement, what are we actually talking about?
Okay, so at its heart, the complement system is this collection, over 50 different proteins. You find them mostly floating in our blood plasma,
Yeah.
but some are also anchored on cell surfaces.
Okay.
And they don't act alone. They work hand in hand with both our innate immune system, the immediate responders, and our adaptive immune system, the more specialized one.
And their main jobs?
Primarily three things: getting rid of harmful pathogens, cleaning up dying or damaged cells, and removing these things called immune complexes, clusters of antibodies stuck to antigens.
It sounds like much more than just a simple defender. And the story of how it was first figured out is, well, pretty fascinating. Back in the 1890s, right? Jules Bordet.
That's the one, Jules Bordet.
Yeah.
He was experimenting with anti-serum, basically serum loaded with antibodies against the cholera bacteria, Vibrio cholerae. And he saw that this anti-serum could make the bacteria burst open, lyse them.
But only if the serum wasn't heated first.
Exactly. If he heated the serum, the lytic power vanished. But, and this was the key insight, if he added fresh serum, even serum without any specific antibodies against cholera,
Or the lysis.
he did. And Ehrlich was the one who actually coined the term complement. He defined it as the activity of blood serum that completes the action of antibody.
A neat definition.
It was a huge step. But here's the interesting part. Initially, because it worked with antibodies, people thought of complement as mainly part of the adaptive immune system. But now, we know its ability to, punch holes in membranes is just one small part of its job.
Yeah, the sheer number of ways microbes have evolved to dodge complement tells you how vital and versatile it really is.
Absolutely. And the whole system, it isn't just randomly floating around. Most of these complement proteins are made primarily in the liver by hepatocytes.
Okay, the liver's the main factory.
Yeah, but not the only one. Other cells chip in too. Blood monocytes, tissue macrophages, fibroblasts, even the cells lining our gut and urinary tracts make some. All told, these proteins make up a pretty big chunk, maybe 15%, of the globulin protein fraction in our blood.
And it's not just what's in the blood, right? You mentioned some are on cell surfaces.
Exactly. A lot of the regulatory components, the ones that keep the system in check, actually sit right there on the surfaces of our own cells. So the system's influence reaches way beyond just the plasma. It's protecting our tissues directly.
Makes sense. So to help us wrap our heads around this complexity, these, 50-plus proteins, they can be grouped into functional categories.
Yeah, about 7 main functional categories. It helps break it down.
Okay, let's walk through them. First up.
First, you've got the initiator complement components. These are the ones that get the whole process started. They bind to a target, maybe directly on a pathogen, maybe via an antibody, and that binding triggers them to change shape and become active. Think C1 key or mannose binding lectin, MBL. Those are key initiators.
Okay, initiators kick it off.
Then you have the enzymatic mediators. Many complement proteins start off inactive, like little molecular time bombs called zymogens. They need to be cut or cleaved by an enzyme upstream to become active proteases themselves.
Proteases being enzymes that cut other proteins.
Right, and these newly activated proteases then cleave and activate the next protein in the chain. The really critical ones here are the enzyme complexes called C3 convertases and C5 convertases. They cut C3 and C5, respectively, unleashing the active fragments. This whole sequence, initiator to effector, that's the complement cascade.
Got it. Cascade. Next category.
Next are the phagocytosis-enhancing components. We usually call these opsonins. When the cascade is running, larger fragments of proteins like C3 and C4, specifically C3b and C4b, get covalently attached, like glued onto the microbes.
They tag them.
Exactly. They act like molecular eat-me tags. They label the invaders, making them way more appealing to our phagocytic cells, the immune cells that engulf stuff, because these phagocytes have specific receptors that grab onto C3d and C4b.
Okay, so the big fragments tag. What about the smaller ones that get cut off?
Oh, good question. Those smaller fragments play a huge role, too, as inflammatory mediators or anaphylatoxins. Think C3a and C5a. These little guys bind to receptors in the cells lining our blood vessels, the endothelial cells.
And what does that do?
It makes the blood vessels wider and leakier. This increases blood flow and lets more immune cells squeeze out of the blood and get into the tissue where the infection or damage is. The name anaphylatoxin actually hints at their power. They can trigger responses similar to severe allergic reactions. Anaphylaxis, if they're not controlled, shows how potent they are.
Wow. Okay, number 5.
Number 5 involves the membrane attack proteins. These assemble into the famous MAC, the membrane attack complex. This is complement's direct kill mechanism.
The hole punching part.
That's the one. These proteins, C5b through C9, insert themselves right into the microbial cell membrane. They form a ring-like structure, basically punching a large hole or pore through the membrane.
And that kills the microbe.
Yep. It destroys the cell's integrity, water rushes in, and boom, the cell excites bursts open. Now, it's mainly aimed at microbes, but if our own regulatory systems fail, the MAC can sometimes form on our own infected cells too.
Right, regulation is key. Okay, category 6.
Yeah. Complement needs to talk to other cells, so we have complement receptor proteins. These are molecules sitting on the surfaces of our various host cells. They act like docking stations, binding to specific complement proteins and triggering specific functions inside that cell.
Like, what kind of functions?
Well, for example, a receptor called CR1 binds to complement-coated pathogens and signals phagocytes to engulf them. Or receptors for C5a on neutrophils tell them to release their inflammatory cargo. They're the communication links.
Makes sense. And the final category, number 7.
And arguably one of the most crucial, regulatory complement components.
Yes.
Our bodies have to control this system. It's so powerful, it could easily damage our own healthy tissues. These regulatory proteins act as the brakes.
How do they do that?
They limit complement's effects, protect our own cells. Examples include factor I, which actually degrades key complement fragments like C3b, and CD59, also called protectin, which specifically stops the MAC from forming on our own cells.
Okay, so bringing all 7 of those together, what's the big picture? What does this whole setup tell us?
It tells us that complement isn't just one thing. It's this incredibly organized, multi-layered system. It's like an orchestra of proteins, each playing a specific part. It's designed for immense destructive power against threats, but also for incredible precision to avoid harming the host. It's really a testament to the sophistication of our immune system.
Okay, fantastic foundation. Now let's get into the nitty-gritty of how this powerhouse actually gets switched on, the activation pathways. There are 3 main ones, right?
That's right. 3 major routes: the classical pathway, the lectin pathway, and the alternative pathway.
Yeah.
But here's the key principle. Despite their different starting points, they all ultimately converge on the same critical step.
Which is?
Generating enzyme complexes that cleave that central protein, C3. These are the C3 convertases.
Okay, so different paths leading to C3 cleavage.
Exactly. The classical and lectin pathways use a C3 convertase made of 2 protein fragments, C4b and C2a, so we call it C4b2a. The alternative pathway uses a different combo, C3b and bambo, so C3bbb. But the result is the same. They slice C3, generating the crucial C3b fragment and the inflammatory C3a.
And it doesn't stop at C3, right? There's another conversion step.
Correct. The next major checkpoint is generating the C5 convertases. These are formed when another C3b molecule binds to the existing C3 convertase. So for classical lectin, you get C4b2a3b. For the alternative, you get C3b-BBC3b.
And these C5 convertases then cleave C5.
Precisely. They cut C5 into C5a, another potent inflammatory signal in C5b.
Yeah.
And C5b is the piece that kicks off the assembly of that hole-punching membrane attack complex, the MAC.
Okay, let's tackle the classical pathway first. This one's interesting because it's often linked with the adaptive immune system, specifically with antibodies.
That's its main trigger, yes. It typically gets initiated when antibodies, specifically IgM or certain subclasses of IgG, bind to their target antigen. This target could be on a microbe, like a bacterium or virus, or part of a soluble immune complex floating around.
And the antibody binding isn't enough, right? The antibody itself has to change somehow.
Exactly. When the antibody binds the antigen, its tail region, the FUC portion, undergoes a subtle but critical shape change. This shape change exposes a binding site for the very first component of the classical pathway, the C1 complex.
C1, what's that made of?
C1 is this multi-part molecule. I have a recognition unit called C1q, which looks a bit like a bunch of tulips, with 6 heads that can grab onto those bound antibody FCE regions. Then it has 2 enzyme components, C1r and C1s, associated with it.
So C1q senses the antibodies.
Right. It needs to bind to multiple FC regions close together to get a strong enough signal. Once C1q locks onto enough antibodies, it triggers a change in C1r and C1s, waking them up, activating their enzymatic activity.
It needs a specific arrangement.
Yeah. IgM is a big pentamer, like 5 antibodies joined together. When it binds to a surface, it flattens out into this staple shape, revealing multiple C1q binding sites all at once. Very efficient.
And IgG, it's smaller.
IgG is smaller, just a single Y shape. So for IgG to trigger C1 efficiently, multiple IgG molecules usually need to bind close together on the target surface, forming little clusters or hexamers. This clustering brings enough FC regions together to bind C1q tightly and kick off the cascade.
But C1q isn't only about antibodies, is it? I remember reading it can bind other things directly.
That's a really important point, yes. C1q has more ancient roles, too. It can directly bind to things like C-reactive protein CRP, when CRP is stuck to bacteria. And maybe more significantly, it can bind directly to molecules exposed on our own dying cells, things like DNA, annexins, histones that appear on the surface during apoptosis, programmed cell death.
So it helps clean up our own cellular debris, too.
Exactly. By binding these apoptotic cells, C1q flags them for removal, preventing them from potentially triggering autoimmunity. We'll definitely touch on this again later, especially with the nervous system connection.
Okay, so C1q binds, C1r and C1s get activated. What happens next in this classical cascade?
Once C1q binding triggers it, one of the C1r enzymes activates itself, then it activates the other C1r. These 2 active C1r enzymes then cleave and activate the 2 C1s enzymes. Now you have fully active C1s protease ready to go.
And what does active C1s cut?
It has 2 main targets, C4 and C2. It cleaves C4 first, releasing a small fragment, C4a, and leaving the larger C4b fragment. This C4b piece has a very short-lived, highly reactive internal bond, a thioester bond.
Thioester. Sounds reactive.
Extremely. When C1s cut C4, this bond is exposed, and C4b tries to immediately form a covalent link, basically gluing itself onto a nearby protein or carbohydrate on the target cell surface. But it has to be fast. If it doesn't find a target within milliseconds, water attacks the bond, and C4b becomes inactive.
So some C4b successfully sticks to the target membrane. Then what?
Once C4b is securely attached, it acts like a docking site for C2. C2 binds to this membrane-bound C4b. Now, the C1's enzyme comes along again and cleaves the bound C2, releasing a smaller fragment C2b, and leaving the active C2a fragment still attached to C4b. This combined C4b2a complex is the C3 convertase of the classical pathway.
C4b2a, and the C3 convertase is where the amplification really happens.
Huge amplification. This C4b2a enzyme is a potent protease. It can now rapidly cleave hundreds, even thousands, of C3 molecules. Each C3 molecule is cut into the small C3a fragment, that inflammatory anaphylotoxin, and the larger, incredibly important C3b fragment.
C3b seems like the star player here.
It absolutely is. Generating lots of C3b is central to almost everything complement does downstream. Just to emphasize its importance, people with the genetic deficiencies in components before C3 cleavage are extremely vulnerable to infections and autoimmune issues. Deficiencies after C3 are generally less severe.
Why is C3b so critical? What does it do?
It has multiple crucial jobs. One, it acts as that opsonin we talked about. It covalently binds all over the microbial surface, tagging it for phagocytosis. 2, it tags immune complexes for clearance. And 3, some of the C3b molecules bind to the C4b2a convertase itself, forming a new complex, C4b2a3b.
And that's the C5 convertase.
That is the C5 convertase of the classical pathway, and its job is to cleave C5, initiating MSC formation. So you see, C3b is right at the crossroads, linking the initial activation to both opsonisation and the final ilytic pathway. Its central role can't be overstated.
Got it. C3b is the linchpin. Now let's shift gears to the lectin pathway. This one is also innate, antibody independent, like the alternative pathway you mentioned.
That's right. The lectin pathway is another way our innate immune system can trigger complement without needing antibodies. Like the classical pathway, it also generates that same C4b2a C3 convertase.
But it starts differently. No antibodies involved.
No antibodies. Instead, it uses proteins called lectins. Lectins are specialized proteins that recognize and bind to specific patterns of carbohydrates or sugars.
And the main one here is MBL, mannose binding lectin.
MBL is the classic example, yes. It specifically recognizes repeating patterns of mannose sugar residues and also things like N-acetylglucosamine, glucose, fucosugars, commonly found on the surfaces of a wide range of microbes.
Like which microbe?
Bacteria like Salmonella, Listeria, Neisseria, fungi like Cryptococcus, Candida, even some viruses like HIV and RSV. MBL acts as a pattern recognition receptor, spotting these common microbial signatures.
And that's clinically relevant, MBL levels.
Very much so. People with naturally low levels of MBL are often more prone to serious bacterial infections, especially as children. And there are other lectins too, like collectins and ficolins, that work in similar ways.
So MBL spots the sugars on a microbe and binds. How does that trigger the cascade? It doesn't have C1R and C1s like the C1 complex.
Good point. MBL circulates in the blood, already associated with its own set of serine proteases, called MBL-associated serine proteases, or MASPs. The main one is MASP2.
Ah, so it brings its own enzymes.
Exactly. When MBL binds to the microbial surface, this triggers the activation of the associated MASP2 enzyme. And activated MASP2 does exactly what C1s does in the classical pathway. It cleaves C4 and C2,
Forming C4b and C2a.
which then assemble into the C4b2a C3 convertase, identical to the one formed by the classical pathway.
And from that point, the lectin pathway just merges with the classical pathway's downstream steps.
Precisely. Once that C3 convertase is formed, everything else is the same. Formation of the C5 convertase, C4b2a3b, cleavage of C5, and assembly of the MAC. It's a beautiful example of convergence. The soluble lectin, MBL, basically substitutes for the antibody of recognition, and the MASP enzyme substitute for C1 or C1s, in cleaving C4 and C2. Different start, same powerful result.
Okay, now for the third route, the alternative pathway. Also antibody independent, part of innate immunity. But you said this one uses different convertase.
That's a key distinction, yes. The alternative pathway uses C3BB as its C3 convertase and C3BBCCB as its C5 convertase. And what's really interesting is that we now understand it can be initiated in 3 different ways.
3 ways. Okay, what's the first one? The tick-over pathway. What does tick-over mean?
Tick-over refers to the fact that there's a constant low level of spontaneous C3 activation happening in our blood all the time. That reactive thioester bond inside C3 isn't perfectly stable. Occasionally, it spontaneously breaks and reacts with a water molecule.
Creating a modified C3.
Exactly. It forms C3H2O. This subtle change allows C3H2O to bind another serum protein called factor B.
And factor B binding sets it up for
Cleavage by factor D. Factor D is another protease always present in the blood. It cuts the bound factor B, releasing a small fragment BA and leaving the larger active fragment BAB still attached to C3H2O. The C3H2OB complex is a C3 convertase, but it's floating freely in the plasma, a fluid phase C3 convertase.
Okay, so there's this constant low hum of C3 activation happening. How does that target actual microbes?
Well, this fluid phase C3H2OB is pretty unstable and gets quickly inactivated in healthy conditions. But it does generate a small amount of C3b. If there's a microbe nearby, some of this newly generated C3b can covalently attach to the microbial surface via that reactive thioester bond.
Ah, so the C3b lands on the microbe.
Right, and this is crucial. Factor B can bind not only to C3H2O, but also to this surface-bound C3b. Once factor B binds to the C3b on the microbe, factor D comes along again, cleaves factor B, and now you have C3BB formed directly on the microbial surface.
And this surface-bound C3BB is the main alternative pathway C3 convertase.
Yes, this is the workhorse. But there's another twist. This C3BBB complex is also inherently unstable. It falls apart quickly.
Propertin gets involved.
Exactly. Propertin, also called factor P, is a positive regulator. It binds to the C3BB complex on the microbial surface and stabilizes it, dramatically increasing its lifespan and activity.
So propertin keeps the enzyme working longer.
Much longer. And this propertin-stabilized C3BB now rapidly churns out loads more C3b right there on the microbial surface. Many of these new C3b molecules then bind more factor B, get cleaved by factor D, creating even more C3BB convertases. It's a powerful amplification loop.
So powerful.
Incredibly powerful. Once kicked off, you can get over 2 million C3b molecules deposited on a bug surface in less than 5 minutes. And here's a key point. Regardless of which pathways started the complement activation, classical, lectin, or tickover, it's estimated that up to 80–90% of the total C3b deposited on a target is actually generated via this alternative pathway amplification loop. It's the main engine of C3b production once things get going.
Wow. So the alternative pathway really turbocharges the whole response, and this constant tick over, this always-on potential.
Yeah.
Makes you wonder how scientists even figured this pathway out, and how do we control it? Which brings us to that Propertin story you mentioned earlier, the tragic one.
Ah, yes. Luby Pilmer. It's a really compelling story and, sadly, quite tragic. Pilmer was a brilliant biochemist, originally from South Africa, did amazing work in the U.S., like purifying tetanus and diphtheria toxins, which was vital for vaccines.
But he got interested in complement.
Deeply. He was fascinated by earlier findings that mixing human serum with zymosin, a component from yeast cell walls, somehow made C3 disappear from the serum. Initially, people thought the C3 was just getting stuck to the zymosin.
But Pilmer dug deeper.
He did, systematically. He showed C3 wasn't just sticking, it was being actively cleaved, broken down, but only under specific conditions: body temperature, neutral pH. That smelled like an enzyme at work.
How did he prove it was enzymatic?
His experiments were elegant. He mixed serum and zymosin in the cold, no C3 cleavage. Warmed it up, cleavage happened. Then, crucially, he mixed them cold, removed the zymosin, and then warmed the remaining serum. Nothing happened. This proved that whatever was cleaving C3 had to be physically absorbed onto the zymosin.
So something from the serum was binding the zymosin and then acting as the enzyme.
Exactly. He figured out it was a specific serum factor. He painstakingly purified this factor, showed it needed magnesium ions, and named it Propertin, from the Latin perdere, meaning to destroy. He linked it to various antibacterial actions.
And what he'd found, we now know, was Propertin stabilizing the alternative pathway C3 convertase, C3BBB, on the zymosin surface.
Precisely. His work was a huge deal, even at the popular press.
Mm.
But then, the challenge came.
Right, Robert Nelson in 1957.
Yes. Nelson argued Pilmer hadn't found anything new. He claimed Propertin was just a mix of natural antibodies that happened to bind zymosin, and Pilmer had simply rediscovered the classical pathway triggered by these antibodies. And sensitive tests did find tiny amounts of anti-zymosin antibodies in Pilmer's prep.
Which cast doubt on Propertin's unique role.
It did, widely. And for Pilmer, who was apparently a very intense and perhaps sensitive person, this public scientific battle was devastating. His reputation was questioned, his findings dismissed by many.
And
Tragically, he died in 1957 from a barbiturate overdose, ruled a suicide. It's a real low point in the history of immunology. A brilliant mind lost under immense pressure.
Just awful. But his work was eventually proven correct.
Thankfully, yes, posthumously. Later research definitively showed you could remove all the anti-zymosin antibodies, and the Propertin prep still worked. They also identified the other key component Pilmer had noted, the heat labile factor, as a distinct protein, factor B. By the late 60s, the alternative pathway, with Propertin as its key stabilizer, was firmly established. A vindication, but far too late for Pilmer. It's a sobering reminder of how science progresses sometimes.
Absolutely. Okay, returning to the alternative pathways initiation, we covered the tick over. You also mentioned Propertin itself can initiate things directly, the Propertin-activated pathway.
Yes, this is more recent understanding. Beyond just stabilizing C3b-bial, Propertin itself can act as an initiator, a recognition molecule. Experiments show Propertin can bind directly to certain microbial surfaces, like chlamydia pneumonia, and also to our own dying apoptotic or necrotic cells.
And once it binds?
Once bound, it seems Propertin can directly recruit C3b, perhaps the low levels generated by tickover, and factor B. This allows factor D to cleave factor B, forming a C3-PBB complex and active C3 convertase right there where Propertin first landed.
So it offers more specific targeting than just random tickover.
Exactly. It provides a way to focus the alternative pathway initiation onto specific surfaces recognized by Propertin, adding a layer of selectivity.
Okay, tickover, Propertin activated. What's the third way? The protease activated pathway. This links complement to blood clotting.
This connection is really fascinating. If you think about it, complement activation and blood coagulation share similarities. Both involve cascades of protease enzymes cutting and activating the next protein. Both have amplification loops.
Makes sense.
And decades ago, scientists found that key clotting factors, like thrombin and plasmin, could directly cleave C3 and C5 in a test tube, releasing those inflammatory bits C3a and C5a.
But was that relevant in the body, or just a test tube thing?
Initially, they thought you needed impractically high thrombin levels. But more recent studies using disease models in mice show that yes, activating the coagulation cascade can lead to physiologically significant cleavage of C5, generating C5a. For instance, in lung inflammation models, thrombin was shown to make C5a even in mice lacking C3, meaning the normal C5 convertase couldn't form.
So clotting itself can directly fuel inflammation via C5a.
It appears so. It adds another layer of inflammatory amplification when clotting is triggered. Probably doesn't generate enough C5b to form much MAC, but the C5a production seems real.
And platelets are involved too, activated platelets.
Yes, another link. When platelets get activated during clotting, they release stuff like ATP, calcium, and certain enzymes kinases. These kinases can phosphorylate C3b that lands on cell surfaces, and phosphorylated C3b is actually more resistant to being broken down by our regulatory proteins.
So clotting helps C3b stick around longer.
Effectively, yes. By making C3b harder to degrade, activated platelets can enhance the activity of all the complement pathways. It's this really intricate crosstalk between different protective systems.
Okay, so we've got these pathways fired up, C3b being generated like crazy, C5 getting cleaved. What are the actual downstream effects, the effector functions of complement? It all starts converging on that C5 convertase and MAC formation.
That's the endpoint for the VLEC pathway, yes. Whether it's a classic lectin C5 convertase, C4b2a3b, or the alternative one, C3b-bbc3b, they both do the same job. They precisely cut C5 into C5a, the anaphylatoxin, and C5b.
And C5b is the key to the MAC.
C5b is the initiator. It's quite unstable on its own, though. It needs to quickly bind to another complement protein, C6, to become stable enough to attach to the target cell surface or immune complex.
And then comes the MAC-induced cell death, the famous hole-punching action.
This is complement's direct lethal weapon. The MAC components C5b, C6, C7, C8, and then multiple copies of C9 assemble step by step right on the microbial membrane.
Walk us through the assembly.
Okay, C5b binds C6. Then C7 joins the C5b-C6 complex. This binding causes C7 to undergo a big shape change, exposing hydrophobic fatty regions that allow it to insert itself into the lipid bilayer of the target membrane. That C7 insertion is the real trigger for the rest of the MAC assembly.
C7 anchors it. Then what?
Next, C8 binds. C8 itself has multiple parts. One part inserts into the membrane, creating a small initial pore, maybe 10 angstroms across. But the real pore comes next.
With C9?
With C9. Anywhere from 10 to maybe 19 molecules of C9 bind to the C5b8 complex. As they bind, they polymerize, arranging themselves into a large, hollow tube that inserts fully into the membrane. This forms the final MAC pore, which is much larger, 70 to 100 angstroms wide.
And that large pore is lethal.
Absolutely. It completely destroys the cell's ability to control what goes in and out of osmotic integrity. Water floods in, the cell swells uncontrollably, and eventually it bursts. Lysis. Death. And remember, while it's targeted at microbes, if our own cells' defenses are overwhelmed, this can happen to infected host cells too.
What about our own nucleated cells? Can they survive a MAG attack?
Red blood cells? No, they lyse easily. But nucleated cells are tougher. Even smaller MC pores, maybe with just a few C9s, can trigger a form of programmed cell death called apoptotic necrosis. It involves calcium rushing in, the nucleus fragmenting. Our cells do have defenses, regulatory proteins like CD59, but they can be overcome.
Can cells fight back or repair the damage?
Sometimes, yes. Cells aren't entirely passive. They can sometimes shed little bubbles of membrane vesicles containing the MACs, effectively ejecting them. Or they can internalize the MAC complex into lysosomes, like cellular garbage disposals, to degrade it. They can try to repair the breach.
And you mentioned a kind of dark side to MACs that aren't quite lethal, sublytic MACs.
Yes, this is really intriguing and still being researched heavily. It seems that if you get just a few MECs forming, not enough to cause lysis, they can sometimes trigger unexpected signals inside the cell. Things like promoting cell division or even inhibiting normal programmed cell death, apoptosis.
That seems counterintuitive for a killing machine.
It really does. It highlights how complex these pathways are and that messing with them, say in cancer therapy, could have unforeseen consequences. A system designed to kill might under certain conditions actually helps cells survive. It's fascinating.
Now, microbial susceptibility. Is everything equally vulnerable to the MNA?
Definitely not. It's an arms race, remember. Enveloped viruses like herpes, influenza, HIV are generally susceptible. Their outer envelope is vulnerable. Gram-negative bacteria are also usually susceptible. The MAC forms at junctions between their inner and outer membranes and breaches both. This is crucial for controlling bacteria like Neisseria meningitidis, the meningitis bug. People lacking MSC components are incredibly vulnerable to Neisseria.
But gram-positives are tougher.
Much tougher, generally. Their very thick peptidoglycan cell wall acts like armor, physically blocking the MVAC components from reaching the delicate cell membrane underneath. So gram-positives are often resistant to direct lysis.
And there are even newer roles being found for these sublytic MACs, something about inflammasomes.
Yes, that's cutting-edge stuff. It looks like even sublytic MAC formation, by letting a little calcium leak into the cell, can trigger the assembly of intracellular protein complexes called inflammasomes. This, in turn, leads to the production and release of potent inflammatory cytokines like IL-1, further amplifying the inflammatory response. So the ME isn't just about lysis, it's also signaling danger.
Okay, beyond direct killing by MA, you said opsonisation. Tagging for destruction might be complement's most important job physiologically.
Many immunologists would argue that yes, opsonisation is simply the process of coating a target, a microbe, an immune complex with these molecular tags, primarily C3b and P4b. Once coated, the target becomes vastly more efficiently recognized and eaten by phagocytic cells.
The eat me signal again.
Exactly. Phagocytes, like macrophages and neutrophils, are studded with receptors, like CR1, CIGNR1, C1QRP, that specifically bind to these complement tags, C3b, C4b, etc. Binding triggers phagocytosis, the engulfment and destruction of the tagged particle.
And red blood cells use this too, for immune complex clearance.
Critically important role. Red blood cells have CR1 receptors. They bind C3b-coated immune complexes circulating in the blood and act like taxis, transporting them to the liver and spleen. There, specialized macrophages strip off and destroy the complexes, preventing them from lodging in tissues and causing damage like kidney inflammation or vasculitis. It's a vital cleanup service.
And opsonization helps fight viruses too, not just by lysis.
Definitely. A thick coat of antibody and complement around a virus can physically block it from binding to and infecting host cells. It's called neutralization. Plus, the complement coat strongly promotes the phagocytosis and destruction of the virus by macrophages.
And there was that other twist. Even if a coated virus gets inside the cell.
Right. Recent work showed that if complement-coated viruses, like certain adenoviruses, somehow end up in the cell's main compartment, the cytosol, they can actually trigger the cell's internal protein disposal system, the proteasome, to degrade them. And it also flips on cellular alarm bells, activating transcription factors like NFB and IRFs, leading to the production of antiviral proteins. It's defense in depth.
And let's not forget those small fragments, C3a and C5a, their role in promotion of inflammation.
Hugely important. These anaphylatoxins, C3a and C5a, are powerful signaling molecules. They bind to specific receptors, C3IR and C5R, which are G protein-coupled receptors found on many immune cells, granulocytes, monocytes, macrophages, mast cells, dendritic cells, endothelial cells.
And what effects do they trigger?
A whole suite of pro-inflammatory actions. They cause cells to release other inflammatory mediators like IL-6 and TNF-A. They make local blood vessels leaky, helping immune cells get to the site. They increase smooth muscle contraction. They directly stimulate phagocytosis. They cause granulocytes like mast cells and neutrophils to degranulate, releasing histamine and other potent chemicals. They can even help guide lymphocytes to lymph nodes to get activated. And systemically, they contribute to fever, which can hinder microbial growth.
So connecting the dots, these small fragments are like broadcasting an emergency signal, rallying the troops, directing traffic, and turning up the heat on the invaders.
That's a great way to put it. They are potent coordinators of the initial inflammatory battle.
Now let's explore how complement acts at the interface between innate and adaptive immunities. This is where it gets really intricate, showing how deeply woven into the whole immune system it is.
Absolutely. This is where we see complement isn't just a standalone killer system, it's a crucial communicator and modulator. A lot of its diverse effects happen because complement fragments bind to specific receptors on various host cells. And the number of these receptors isn't fixed. Cells can ramp up their expression, like phagocytes increasing complement receptors tenfold when activated.
So these complement receptors are the key connectors. What are some of the most important ones bridging innate and adaptive immunity?
Okay, let's highlight a few key players. CR1, or CD35, is a big one, found on red blood cells, phagocytes, B cells, and others. It binds C3b and C4b mainly. On red cells, it clears immune complexes. On phagocytes, it boosts engulfment. On B cells, it helps them grab and present antigens to T cells. Plus, it has regulatory roles in C3 breakdown. Very versatile.
Then there's CR2, CD21. You mentioned this is critical for B cells, the antibody makers.
Hugely critical for B cells. CR2 is mostly on B cells and follicular dendritic cells, FDCs. in lymph nodes. It binds to break down products of C3b, like C3 and IC3b.
Yeah.
Now, here's the magic. If an antigen is coated with these complement fragments and it binds simultaneously to the B cell's main antigen receptor, the BCR, into CR2,
It turbocharges the B cell activation.
radically.
Yeah.
It acts as a co-receptor, lowering the amount of antigen needed to trigger a strong B cell response by maybe 100-fold. It makes B cells super sensitive to complement tagged threats. And yes, problems with CR2 are linked to autoimmune diseases like lupus, SLE.
What about receptors on phagocytes like macrophages?
Besides CR1, they heavily rely on CR3, CD11b CD18, and CR4 CD11c CD18. These are found on monocytes, macrophages, neutrophils, even NK cells. They primarily bind IC3b, another C3b breakdown product, strongly promoting opsonisation and phagocytosis. CR3 also binds the regulator factor H. Then there's C-Reg, mostly on liver macrophages. It binds C3b and IC3b and is vital for clearing stuff from the blood. Mice lacking CRIG do worse with infections.
And C1QRP and CTNR1, what do they do?
C1QRP, CD93, binds C1q and MBL, enhancing phagocytosis. It's on various cells. SAGNR1, CD209, is interesting. It's on macrophages in the spleen and lymph nodes. It's a lectin that binds C1q and carbohydrates directly, like those on Streptococcus pneumonia. Binding activates its C1q grabbing ability, leading to opsonization and handing off the bacteria to other immune cells. This helps explain why people without a spleen are so vulnerable to S. pneumonia.
And the receptors for the anaphylatoxins C3A and C5A.
Those are C3IR and C5R, CD88. They're the GPCRs we mentioned, found on mast cells, granulocytes, etc., mediating all those inflammatory effects. There's also C5L2, which binds C5A but seems to act more like a brake, dampening the C5A signal. Animals without C5L2 have exaggerated inflammation.
Okay, let's process this. The sheer variety of these receptors on so many different immune cell types, it really paints a picture of complement as this intricate integrator, weaving together different parts of the immune response.
It's a master coordinator, absolutely. And you see this clearly in its impact on antigen-presenting cells, APCs. Dendritic cells, FDCs, macrophages, they all express many complement receptors.
How does complement binding affect them?
When complement components like MBL, C1q, C3b, or C4b bind to an antigen, it significantly enhances the uptake of that antigen by APCs. Better uptake means better processing and presentation to T cells, leading to a stronger adaptive response.
And C5A signaling on APCs.
C5a, binding to C5r on APCs, can influence their migration patterns and what kind of cytokines they produce. For instance, it can boost production of IL-12, which tends to push the T-cell response towards a Th1 type good for fighting intracellular pathogens. It's a fine-tuning mechanism.
This influence clearly extends to B-cell-mediated humoral immunity, too, building on that CR2 co-receptor idea.
Definitely. Early studies showed if you deplete C3 in mice, their antibody responses suffer badly, especially T-dependent ones. That CR2 co-receptor function is key, lowering the activation threshold, making B cells respond more readily to complement-tagged antigens. It links the innate tagging by complement directly to adaptive antibody production.
And even T-cell-mediated immunity is impacted. This was surprising to me. There's that fascinating quality control check on young T cells.
Yes, it's really elegant. So we have these natural antibodies, mostly IgM, circulating around, binding common patterns. As T cells mature in the thymus, they're supposed to get coated in sialic acid, a type of sugar. This coating acts like a shield,
Protecting them from what?
protecting these newly released T cells, called RTEs, mesenchymic emigrants, from being accidentally targeted by those natural IgM antibodies and subsequent complement lysis. The sialic acid also helps them bind regulatory factors like factor H.
So if a T cell doesn't get enough sialic acid,
It's curtains for that T cell. If an RTE leaves the thymus with defective sialylation, it's highly vulnerable. Natural IgM binds, complement gets activated, and because the cell can't properly express inhibitors like CD55 or bind factor H, the complement cascade runs to completion and the cell is destroyed.
so complement acts like a bouncer for the T cell pool.
A quality control checkpoint, exactly, ensuring only properly matured, protected T cells make it out into circulation.
And for mature T cells, complement isn't just destructive, it helps them survive and function.
Right, it's not just about killing, it's quite paradoxical. Th1 cells, a type of helper T cell, actually need to produce low levels of C3a and C3b inside the cell for their own survival. And when T cells get activated by their antigen, they secrete C3a, C3b. The C3a then binds back to receptors on the T cell itself, boosting pro-inflammatory cytokine production that supports the Th1 response.
And C3b helps T cell growth.
Yes, interaction between C3b fragments and another molecule on the T cell surface, CD46, seems important for Th1 growth and survival.
And it even helps shut down the response later.
It seems so. During the contraction phase, sustained complement production by T cells might induce IL-10, a regulatory cytokine that helps dampen the Th1 response and promote apoptosis of excess T cells. Plus, C5a can enhance the activation of CD8 plus killer T cells, possibly by boosting IL-12 from APCs. So the take-home is, complement is deeply involved in regulating T cell life, death, and function. It's a critical bridge, modulating both B and T cell immunity.
Truly remarkable. And its roles go even further beyond fighting infection into processes like immune contraction and even nervous system development.
This is where our view of complement is really expanded. That contraction phase after an immune response is vital. You've got all these extra lymphocytes that need to die off cleanly via apoptosis, and you've got leftover immune complexes. Both need safe disposal to prevent autoimmunity. Complement is essential for both.
How does it help with disposal of apoptotic cells?
When cells undergo apoptosis, they flip a lipid called phosphatidylserine to their outer surface. It's an eat-me signal. This signal gets recognized by C1q, either directly or via bridging proteins like annexin A5. Dying cells also expose DNA, which C1q also binds.
And C1q binding flags them for removal.
Exactly. C1q binding strongly promotes phagocytosis of these apoptotic cells and the little membrane blebs they shed, called apoptotic bodies. This clearance can happen directly via C1q receptors on phagocytes, or indirectly by C1q initiating the classical pathway, leading to C3d deposition, which then triggers phagocytosis via CR1.
And this is clinically important. C1q deficiency.
Hugely. Mice lacking C1q get autoimmune diseases like lupus-like kidney disease, glomerulonephritis, and have higher mortality. Why? Because if apoptotic debris isn't cleared efficiently, it hangs around and can act as self-antigens, triggering the immune system to attack itself.
And it's also critical for disposal of immune complexes, those antibody-antigen clumps.
Absolutely. This is another key anti-autoimmunity function. Circulating immune complexes get coded in C3b. Then, as we mentioned, red blood cells, using their CR1 receptors, pick up these tagged complexes.
The red blood cell taxi service.
Precisely. They transport the complexes to the liver and spleen, where specialized macrophages grab them off the red cells and destroy them. Efficient clearance.
Which is crucial in diseases like SLE, lupus, where complex clearance fails.
Exactly. In SLE, these complexes aren't cleared properly. They deposit in tissues like kidneys and blood vessels, trigger complement locally, and cause inflammation and damage. Now, here's that paradox again. SLE is strongly linked to deficiency in early complement components like C4. About 90% of people lacking C4 get SLE.
Right. How does less complement lead to a disease seemingly caused by complement?
The thinking is that the early complement components like C4 and C1q are essential for that initial C3b tagging and clearance of immune complexes and apoptotic cells. If you lack C4, you don't deposit enough C3b early on. Clearance fails. The complexes and debris persist, deposit in tissues, and then eventually trigger the later, damaging inflammatory and lytic parts of complement, causing the disease pathology. So the early parts are protective via cleanup. Their absence allows the later parts to become destructive.
Fascinating. Complement. Essential for attack, but also essential for cleanup and preventing self-attack. Now, for maybe the most surprising role, advances box 5-2. Complement and the visual system, a brain connection. That blew my mind.
Mine too, when I first learned about it. For ages, complement was purely immunology. But then decades ago, people realized brain cells, specifically microglia, the brain's immune cells, can make complement proteins. But the big breakthrough came in 2007 from Ben Barr's lab. They showed complement proteins are actively involved in wiring the developing brain.
Using the visual system as a model.
Exactly. They look at how nerves from the retina connect to a brain area called the DLGN. In baby mice, DLGN neurons get inputs from both eyes. But in adults, they only get input from the opposite eye.
So the wrong connections have to be removed during development. Synaptic pruning.
Synaptic pruning, precisely. In the first few weeks after birth, the visual system prunes away those inappropriate connections from the same side eye. Barr's team found that during this pruning period, other brain cells called astrocytes release signals, later identified as TGF, that tell the retinal nerve cells, RGCs, to make C1q. And crucially, they saw a C1q protein right at the synapses in the DLGN.
But not at all synapses, only the ones being pruned.
That was the key observation. C1q was found associated with synaptic markers, but not decorating fully formed, healthy synapses. It seemed to be specifically involved in dismantling unwanted connections. The clincher was using knockout mice. Mice lacking C1q or C3 had major defects in this synaptic pruning process. The inappropriate connections persisted into adulthood.
Incredible, but there are still unknowns.
Oh, definitely. We don't know exactly what C1q binds to on the synapse, how the correct synapses are protected, or the full extent of complement's role in pruning across the whole brain. It's clearly involved, but it's not the only mechanism.
And this role in healthy brain development has huge implications for diseases involving inappropriate synaptic loss.
Absolutely. If complement prunes synapses during development, could it be inappropriately reactivated later in life to destroy healthy synapses in disease? Evidence points that way. In glaucoma models, C1q goes up early, and blocking C1q protects the neurons.
And macular degeneration.
Another prime example. Macular degeneration involves buildup of cellular debris under the retina. This debris activates complement, which then damages the crucial retinal support cells, RPE cells. Genetics, imaging, biochemistry, it all points to complement being a major player.
And potentially broader neurodegenerative diseases, too, like Alzheimer's.
That's a huge area of investigation. Complement components are found upregulated in Alzheimer's brains and other neurodegenerative conditions. Researchers are actively exploring whether complement-mediated synapse destruction contributes to the cognitive decline seen in aging and diseases like Alzheimer's. It's completely changed how we think about brain health and disease. A system born in immunology is now central to neuroscience.
Given this immense power, clearing debris, shaping brains, killing microbes, but also potentially damaging our own tissues, regulation of complement activity must be incredibly sophisticated.
It has to be. With those powerful amplification loops and the constant low-level tick-over of the alternative pathway, the system needs tight leashes. The goal is to focus destruction only on pathogens and minimize collateral damage to our own healthy cells. And our bodies use both passive features and active protein regulators to achieve this.
Okay, what are the passive regulatory mechanisms?
A couple of key things. First, simple instability. Many activated components, especially those convertase enzymes like C3-BBAB, just naturally fall apart very quickly. C2-BBAB only lasts about 5 minutes unless Properdin stabilizes it. This short half-life limits runaway activation.
And the second passive mechanism?
It's about the differences between our cell surfaces and microbial surfaces. Our cells are coated in negatively charged molecules, especially sialic acid. These host-specific molecules do two things. They preferentially bind and activate soluble enzymes that destroy C3b, and they also act as docking sites for our soluble regulatory proteins like factor H and C4BP.
So our regulators get concentrated on our own cells.
Exactly. Factor H and C4BP bind to our sialic acid-rich surfaces, ensuring that factor I, the C3b/C4b cleaving enzyme, primarily acts on complement deposited on us, not on microbes, which generally lack these specific host markers.
Clever. Now, what about the specific active regulatory proteins, the dedicated inhibitors?
Right, these are crucial. First, there's C1 inhibitor, C1INH. It's a plasma protein, a type called a serpin. Its main job is to shut down the classical and lectin pathways early. It binds to the active C1R and C1's enzymes, or the MASPs, in the lectin pathway, and causes them to physically detach from the C1q recognition part.
So it stops the initiation.
Yes, it's the only regulator that inhibits the very start of the classical and lectin pathways. It puts a time limit on their activity.
And lacking C1INH causes?
Hereditary angioedema, HAE, that uncontrolled swelling condition due to excessive production of vasoactive peptides because C1 isn't being properly inhibited. It's often inherited as an autosomal dominant trait.
Okay. Then you have decay accelerating factor, DAF, which is CD55, and also CR1, CD35 acting as regulators.
Yes, both DAF and CR1 are anchored in our host cell membranes. Their job is to actively break apart the C3 convertases if they happen to form on our cells. They accelerate the decay.
How do they do that?
For the classic lectin convertase, C4b2a, DAF and CR1 basically prise C2a off C4b, C2a floats away, and the remaining C4b can then be permanently inactivated by factor I. For the alternative convertase, C3bb, they work with factor H to kick Bob off C3b, Bob floats away, and factor I degrades the C3b. Because DAF and CR1 are on our cells, they protect us while leaving convertases on microbes intact.
And factor I seems like the executioner actually cutting up C3b and C4b.
It is. Factor I is a soluble protease always active in the blood.
Yeah.
But, and this is key, it can only cleave C3b and C4b effectively when they are bound to one of those cofactor proteins we mentioned.
Like CR1 or factor H.
Exactly. Or MCP, CD46, on cell surfaces, or C4BP in solution. These cofactors essentially hold C3b or C4b in the right way for factor I to cut them into inactive pieces like IC3b, C3c, C3dg. Because these cofactors are either on our cells or preferentially bind our cells, thanks to sialic acid, factor I activity is targeted to protecting the host.
And factor H variations are linked to macular degeneration.
Strongly linked, yes. Variations that impair factor H function seem to increase risk for AMD, likely due to less effective complement control on retinal cells. And MCP, CD46, has that interesting role in apoptosis. T cells shed it as they start to die, which allows C3b to deposit and flags them for removal. It's elegantly controlled.
How do we stop the MAC, the final hole puncher, from forming on our own cells?
Two main players there, CD59 protectin and S protein, fitronectin. CD59 is anchored in our cell membranes. If a C5b8 complex starts to assemble on our cell, CD59 binds to it and physically blocks C9 from binding and polymerizing. No C9, no big pore.
And S protein.
S protein is soluble in the plasma. It binds to any C5b67 complexes that might form in the fluid phase, preventing them from randomly inserting into bystander host cell membranes. Both CD59 and S protein are crucial guards against self-inflicted amyoglobinuria.
And the clinical link here is PNH, paroxysmal nocturnal hemoglobinuria.
Exactly. PNH is caused by a mutation preventing cells from attaching GPI-anchored proteins, including both DAF, CD55, and CD59. Without these protectors, red blood cells become highly susceptible to complement lysis, leading to the symptoms of PNH. It perfectly illustrates why these regulators are vital.
Finally, how are the inflammatory signals C3a and C5a switched off?
Their activity is very short-lived because of carboxypeptidases, NBAR, circulating in the serum. These enzymes rapidly snip off the terminal arginine amino acid from both C3a and C5a. This converts them into inactive Des-arg forms. Plus, the C5L2 receptor acts as a scavenger for C5a. It's a rapid shutoff system for inflammation.
Which leads to the question, if our system is so powerful and tightly controlled, how on earth do microbes survive? Let's talk microbial complement evasion strategies.
It's a huge topic, and the diversity of strategies is just astounding. It really underscores how critical complement defense is, because microbes have poured so much evolutionary effort into countering it. And most microbes use multiple tricks, not just one.
What are some general tactics? Less specific.
Well, as we mentioned, gram-positive bacteria often rely on their thick cell walls and capsules as physical armor against the MAC. Some bacteria just hide, escaping into intracellular compartments inside host cells, where circulating complement can't reach them. But these are often energy-costly strategies. Many microbes have evolved much more specific, targeted ways to interfere.
Like messing with the antibody-complement link.
That's a common one, especially for viruses. Some viruses make proteins that bind to the epiacy tail of antibodies, physically blocking C1q from docking and starting the classical pathway. Others produce factors that speed up the removal of antibody-antigen complexes from infected cell surfaces or cause them to be internalized rapidly, hiding the target.
Or they just bind and inactivate complement proteins directly.
A very direct approach. Staph aureus, for example, makes a protein called SCIN that specifically binds and inactivates the alternative pathway, C3 convertase, C3bDB. Some parasites, like cystosoma, make a protein that wedges itself between C4b and C2a, preventing the classical C3 convertase from forming properly.
Some just chew up complement proteins with their own enzymes.
Brute force, yeah.
Yes.
Pseudomonas aeruginosa secretes proteases like elastase that can degrade C1q, C3, C3b. Strep bacteria have enzymes, SCPA, SCPB, that specifically target and degrade the C5a anaphylotoxin, dampening inflammation. Some strep toxins even degrade properdin, destabilizing the alternative pathway convertase. Fungi like Aspergillus secrete proteases that chop up C3, C4, C5, C1q, even antibodies. Very dangerous in immunocompromised people.
And perhaps the most insidious strategy, mimicking or stealing our own regulatory proteins.
This is incredibly common and clever, turning our own weapons against us. Many microbes have evolved ways to bind our soluble inhibitors, like factor H or C4BP, directly onto their own surfaces. Strep pyogenes uses its M proteins to recruit factor H and C4BP, which then accelerate the decay of any C3 convertases trying to form on the bacteria.
They can actually steal our membrane-bound ones too, like CD59.
It seems so. Helicobacter pylori, the stomach ulcer bug, has been found coated in CD59. The thinking is it somehow acquires the GPI anchor, and thus the CD59 protein, directly from host stomach cells, using it as a shield against a mass attack.
And viruses must be masters of this mimicry.
Oh yes. Variola, smallpox, and vaccinia viruses actually encode their own proteins that mimic our regulators. These viral proteins bind C3b and C4b and act as cofactors for our factor I, tricking our system into inactivating complement on the viral surface.
Wow. What else?
Some viruses induce the host cell they infect to overproduce our regulators. Others carefully bud out from areas of the host membrane already rich in regulators like CD59, essentially cloaking themselves. Some even incorporate lots of sialic acid into their own viral envelope, mimicking our cell surfaces to recruit factor H and inhibit complement.
So looking at this huge arsenal of evasion tactics, what's the takeaway?
It's a constant dynamic evolutionary battle. The sheer ingenuity microbes display in countering complement tells us just how fundamentally important this system has been and continues to be for our survival over millions of years.
Speaking of millions of years, let's trace the evolutionary origins of the complement system. It's an ancient defender.
It truly is. Although we first understood it through its link to antibodies and lysis, genomic detective work reveals the MAC components, C5b-C9, are actually quite late additions evolutionarily.
So before the MAC existed, what was early complement doing?
The evidence suggests its earliest roles in invertebrates and the first vertebrates were likely focused on opsonisation, tagging things for cleanup, and promoting phagocytosis by primitive immune cells. Think basic recognition and disposal. By analyzing gene families, we can trace the origins of different components back through deep time.
What seems to have appeared first, the oldest parts?
The components of the alternative pathway seem to be the most ancient. Genes for factor B and the C3 family, C3, C4, C5, the ones that get cleaved, appear very early on. We find them in most invertebrate deuterostomes, like sea urchins, and even some protostomes, like horseshoe crabs. Their absence in flies and worms seems to be a later loss, not that they never had them.
Okay, alternative first. Then the lectin pathway components.
Yes, the lectin recognition components, the family, including C1q, MBL, and ficcolins, came next. We find prototype C1q genes in jawless fish like lampreys, and even in invertebrates like sea urchins and tunicates. Crucially, this is before immunoglobulin antibody genes appeared.
So C1q was likely recognizing things like carbohydrates directly before it ever latched onto antibodies.
That's the idea. Early pathogen recognition, antibody independent. MBLs also appear in lampreys. We see functional lectin pathways in tunicates, so that recognition system is very old.
And the classical pathway components, the ones linked to C1q and antibodies.
Those emerged later, mainly through gene duplication events in vertebrates. This gave rise to distinct C2 and factor B, distinct C1 as in MASPs, and distinct C3, C4, and C5 from a common ancestor. The full classical pathway, working closely with antibodies, really solidified with the appearance of jawed vertebrates.
And finally, the membrane attack complex, MAC components, the lytic machinery.
These were the last major addition, appearing around the same time as the adaptive immune system itself. The proteins C6 through C9 all share a specific structural domain, the MACPF domain, suggesting they evolved together for pore formation. We find the full set in mammals, birds, amphibians, even sharks, early jawed vertebrates have complement that can lyse cells and form MAC-like pores.
Are there even more primitive versions of these MAC proteins?
Yes. Ancestral ACPF genes are found even further back, in tunicates and Amphioxus. But these primitive versions seem to lack the key part needed to interact with C5b, suggesting they couldn't form a full C5b-initiated MAC. Interestingly, the proteins structurally most similar to our MAC components are actually toxins found in sea anemones used to punch holes in prey. Shows the deep evolutionary roots of pore-forming weapons.
So, summing up this evolutionary saga,
Complement evolved through diversification and duplication of about 5 core gene families. It started simple recognition and tagging for disposal. Then, as adaptive immunity emerged and the arms race with pathogens intensified, it added layers of complexity, culminating in the sophisticated multi-pathway system with the potent MSC that we see today. It's a living record of host pathogen coevolution.
what an incredible journey we've taken today. We've really gone deep into the world of the complement system. From that initial finding, the heat label component, that completed antibody action.
Yeah, Bourdais' discovery.
To seeing it now as this incredibly sophisticated multitasking system in our bodies.
We've traced those 3 main activation pathways: classical, lectin, alternative. Different triggers, but all funneling down to unleash complement's power. We've seen its incredible range of functions, blasting pathogens with the MA, tagging things for cleanup with opsonins, orchestrating inflammation via anaphylatoxins.
And even those completely unexpected roles, like sculpting synapses in the developing brain.
Right, and we dug into how our bodies keep this beast under control with all those regulators, the checks and balances. And then the flip side, The amazing, devious ways microbes fight back, the evasion strategies. It really highlights complement's importance.
This deep dive makes it crystal clear. Complement isn't just a part of immunity, it's truly foundational. Like you said, a Janus-faced god. Looking back to ancient innate defenses and forward to complex adaptive responses, opsonization, inflammation, lysis, regulation, brain development, it's indispensable.
Absolutely. It touches so many aspects of our physiology.
So as we keep learning more, uncovering new functions for complement, what's the next frontier? Could targeting or maybe even boosting the system revolutionize how we treat neurodegenerative diseases? Or cancer? Or even aging itself? It really feels like the story of complement is still unfolding.